All right, everyone, and this continues our discussion or review for the examination. And this time around, we'll talk about polygenic inheritance. Now, the major difference with other inheritance we talked about, like the codominance, the incomplete dominance, and then the multiple alleles and the Mendelian inheritance, the monohybrid and dihybrid crosses, the major difference is that this previous inheritance that we have discussed only have a few genes, while polygenic inheritance will have many genes, which implies that for the previous discussions that we had, like the incomplete, the codominant, multiple alleles, and the Mendelian inheritance, it has only a few, or they have only a few phenotypes, while polygenic inheritance will have a variety, a great number of variation or great number of, or great variety of phenotypes in between. So this will be the major difference. Let's go straight into that. So polygenes are also called quantitative inheritance and has a continuous variation, as you can see here, as compared to the incomplete dominance, the codominant, multiple alleles, and Mendelian inheritance, which has discontinuous variation because they only have a few counts or a few phenotypes, as compared to polygenes, which had a variety, so many phenotypes that can be found in between. Now, this would also give us more information about continuous and discontinuous variation. So for continuous variation, it has no distinct category, no limit on the value, and tends to be quantitative. It can be measured. So you can measure height, you can measure weight, and you can, to a degree, measure the skin color. So those are examples of continuous variation. Height, weight, heart rate, finger length can also be measured, leaf length can also be measured, and there will be several phenotypes in between the extremes. For this continuous variation, there's no distinct category, there's no in-between category, and that's very important, and tends to be qualitative. So example of these are blood groups, which only have blood type A, blood type B, blood type O, and blood type AB, only 4 of that compared to the range that can be found in height, weight, heart rate, and finger length. Fingerprints also would have discontinuous variation. If representation will be talked about, the continuous variation is represented by a line graph, particularly a bell-shaped curve, while the bar graph can be, while the discontinuous variation can be represented by bar graph with only a few categories. The continuous variation is controlled by a lot of genes and also the environment, so don't forget that. And this continuous variation would only be controlled by a few genes. So it's very interesting that continuous variation, polygenes, are affected by environment. Let's go back to the weight. When we talk about weight, there's a range of weights possible because not only is it controlled by the genes, by your genetic, by the genes that you have inherited by your parents, but also the amount of food that you eat, the kind of nutrition that is found in the food that you eat. So there would be a difference, a great difference in the kind of food and nutrition. So you could expect that there will be a variety, a range of weights that can be possible. That's why this is a characteristic of polygenic inheritance called continuous variation. So that's very interesting. Because continuous, only a few genes would be affecting it. So here's a picture of the continuous variation of great variety of skin color, great variety of eye color, and a great variety of height as well. Other examples that mentioned would be intelligence and behavior. So the phenotypes would be a continuum of uh, that trait. So that's kind of interesting here. Okay, now to understand further, we're still going to be making use of the big A and the small a, the dominant and the recessive, but in this case, there is no really dominance relationship, but rather an additive relationship because the big A is considered to be simply a contributory allele, while the small a is considered to be a non-contributory allele. What do we mean by this? This means that the big A, if it's present, it will contribute to the phenotype. If it's a height, it will add more height. But for the small a, the recessive allele, it is non-contributory, so it doesn't contribute any kind of any kind of or to a 50 degree only a small amount of the phenotype. So that will be the great difference between the two. So again, the big A is contributory, the small a would be non-contributory. Okay, so one more thing you have to take note that the contribution of the dominant all has equal effect for each contributory allele and it contributes to the phenotype. So whether it contributes or not, that will be the characteristic of the big A or the small a, the dominant or the recessive. So this is an example here. You see here all the blues which are non-contributory and all the reds that are contributory. Notice that the color of the color of the eyes would vary from very light going towards the very dark, the other extreme. So gray, blue, green, light brown, and dark brown. And notice that it has a parallelism to the number of contributory allele. For the gray one, it has no contributory allele. So there's very little amount of melanin in the iris, or in the eyes. Here is the blue. There are only two contributory alleles, adding more melanin. For the green one, more melanin is added, so there are four of them. For the light brown, there are more contributory alleles, 1, 2, 3, 4, 5, 6, leading to light brown eye color, while the dark brown would have all of the contributory allele, 1, 2, 3, 4, 5, 6, 7, 8. So that would explain to us that the more dominant letter, the more capital letter that we have, the more of the phenotype we have. Whether it's color, the darker it is. When it's height, the taller it is. When there's more of the capital letters present. So here's an example of a cross. You have here a very dark, all capital, big A, big A, big B, big B, big C, big C. Very light, small a, small a, small b, small b, small c, small c. So the very dark all have the contributory alleles, six alleles, while the very light doesn't have any contributory allele, all recessive over here. So when you cross the very dark with the very light, this would be the whites, for example, in North America, and the very light will be the Negroes in North America. If you cross them, you produce a phenotype that is in the middle, not too dark, not too light. And that is because you will have here only three dominant genes or alleles, big A, big B, and big C, and small a, small b, and small c. So contributory alleles will only be three, leading to a phenotype that is in between, and you call that actually as mulatto, as you will be seeing here. Okay, so this will be mulattoes. That will be the big A, big B, big C, small a, small b, small c. So there are only three. American white would be all recessive. American Negroes will all be has contributory alleles, all dominant alleles over here. Going back here, so we can see that when we have this, we will only have mulattoes. How about if we cross them, the F1 generation with another F1, which means heterozygotes for all three gene pairs would be crossed with an individual with all heterozygote for all gene pairs as well. Then you will have 64 possible combinations, and then you will see here a range of that color. So that is why this is interesting. It will produce a bell-shaped curve with with what we call as changes in the color. So you will see here that for maximum of all dominant, you will have very dark color, but only 5 of the dominant will get dark. Only 4 of the dominant, which means you have 2 recessive, will be fairly dark, and only 3 of this dominant will produce moderate or intermediate skin color. Only 2 dominant and 4 recessive will produce very light color, and 1 dominant and 5 recessive will produce light color, and if you have no dominant, all recessive, you will have very light color of the skin. So this would show us that there is indeed an additive effect for every dominant, every capital letter that you are adding into the genotype. Okay, so we finish with this. The parents will be very dark, very light. You cross them and then you produce the F1 generation. What if you cross 2 F1 generation? Then you will have the F2 generation. And how do you get the F2 generation? Let's make a Punnett square. Let's get the unique gametes of the first F1 generation. So if this is the case, in order for you to get the unique gametes, it's 2 raised to the 3. So that will be 8. So you have 1, 2, 3, 4, 5, 6, 7, 8. 8 possible combination of the gametes for the first individual. For the second F1 generation, it's also 2 raised to the 3. So you have 8 possible gametes. It's the same. 1, 2, 3, 4, 5, 6, 7, 8. So these are 8 possible combinations of the unique gametes. So if you're going to make a Punnett square, on one side, you'll put all of the 8 gametes here. On top, you will put all of the gametes here. And then you'll make some combination, as you can see here. So this will be 1, 2, 3, 4, 5, 6, 7, 8. That's all of the unique gametes of the first F1, and then this will be 1, 2, 3, 4, 5, 6, 7, 8, all of the gametes of the second F1 generation. Now notice, if you combine them together, then you will have the genotype of the F2 generation. So in this case, big A, big B, big C gamete combined with a big A, big B, big C gamete produces a big A, big A, big B, big B, and big C individual, which means since all of these are dominant alleles, 6 of them, then this will be the darkest color. On the other hand, go to the other side, the small a, small b, small c gamete of the first pair of the first F1, and the small a, small b, small c of the second F1. If you combine them together, this should be small a, small a, small a, small b, small b, small a, small c. You all have recessive alleles, and this means this will be the lightest color, and all the possible combinations here is here. So out of the 64, one that looks like a parent is only one that looks like the other parent is only one as well here. And then if you count the other one, then you will be taking note of the possible count. Now, how do you know the phenotypic ratio? Well, actually, you don't need to make the Punnett square anymore, but rather you can make a use of a particular, what we call as the equation. And what's the equation? Let me just go very fast here. You make use of the Pascal's triangle, where the first letter is the dominant and the second letter over here is the recessive, and you raise all of that to 2 raised to the n, where the n would be the number of gene pairs. So let's look at this one. So there's another form of the expression. The small a here would be the dominant, the capital letter. The small b would be the recessive, or the small letter raised to the 2n, where the n would be the number of gene pairs present. So you make use of the binomial expansion. So the a here are all dominants, the b here would be all recessive. So in the first place, if you have 3 gene pairs, like the example that we have a while ago, so this will be 3 gene pairs resulting to 1, 2, 3, 4, 5, 6 alleles. Then you will have here raised to the 6, and that will have a binomial expansion of A6, 6, 15, 20, 15, 6, and then 1. So take note that the phenotypic ratio can be derived from the Pascal's triangle. 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to b. And going here, you see here that if you have 1 gene pair, which means you have 2 alleles over here, then the expression is this one, 1A2, 2AA plus 1A2. So you have a 1 is to 2 is to 1 phenotypic ratio. If you increase this into 3 gene pairs, then you will have, if you have only 1 gene pair, you have 2. If you have 2 gene pairs, that will be resulting to 4 alleles. Then you have a 1 is to 4 is to 6 is to 4 is to 1 phenotypic ratio for 2 gene pairs. If you have 3 gene pairs, which means 6 alleles, then you have a 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1 phenotypic ratio. How do you know the genotypes? Then you just make use of the expansion over here. So for example, we have here 1 is to 2 is to 1. So you have 2 dominant over here, 1 dominant, 1 recessive, and 2 recessive. If you have 2 gene pairs, 1, 4, 6, 4, 1 phenotypic ratio, then you have 4 of this dominant, 3 of this dominant, and then 1 recessive, 2 of this dominant and 2 recessive, 1 of this dominant and 3 recessive, and then all recessive would be over here. So that's how you make use of how do you find the genotype. So let's try to have an example there. Here's an example. Let's go back a little bit. Okay, where is that? Here it is. And okay, you will be noticing a 1, 6, 15, 20, 15, 6, and 1. So the first one would be having uh, all a raised to the 6. So all of this would be recessive. Next one is only one would be dominant, the rest will be recessive. This will have two dominant. So that's all the possible combination. You have three dominants. These are all the possible combination of three dominants. Those are the genotypes. These are four dominants. That's all the possible genotype combination. You have five dominant, big A, big A, big B, big B, big C, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and then all dominant, you only have one of that. So that is how you write down the genotypes. Now there are a few more equations that will be very helpful, and this will be one. If you are asked how many of the F2 generation for polygenes have the same phenotype as the parent, then that answer would be 1 divided by 4 raised to the n. So if you only have one gene pair, that's 1/4. If you have two gene pairs, big A small a, big B small b, then that's 1 of 16. If you have three gene pairs, big A small a, big B small b, big C small c, then you will have 1 out of 64. So that will be the answer. The fraction of F2 similar to the parent. You can also compute for the number of genotypes by making use of the equation 3 raised to the n, where the n would be the number of gene pairs. So 3 raised to the 1 is 3. So there's only 3 genotypes. 3 raised to the 2 gene pairs, you will have 3 times 3. 3 raised to the 3, you have 9. 9 genotypes are present or possible. If you 2 raised to the 2 would be 9 possible genotypes. If you have 3 raised to the 3, you have 3 times 3 times 3, then you have 27 possible genotypes. Okay, so let me just go straight to the example, but just to mention this pleiotrophy, you have a single gene but many traits are produced, just like in sickle cell anemia, where you only have one gene for sickle cell anemia, but you have several possible phenotypes like could cause blindness, but not only that, liver failure and heart attack. And then epistasis, on the other hand, would be there are one gene pair is being affected by another gene pair that may be far away from it. So one locus is masked or is modified by another gene on another locus like this one. Okay, so we won't be talking about that in the examination, just to mention this. Here are some sample problems. Okay, assume that height in a plant is controlled by two gene pairs and that each additive allele contributes 10 centimeters to a base height of 10 centimeters. So you have small a, small a, small b is equal to 10 centimeters. Okay, so let's go to the straight to the question. What is the height of an big A, big A, big B, big B plant? So just take note of the clue. Every dominant gene, every dominant allele has an additive effect and it contributes 10. So you have 1, 2, 3, 4. There are 4 dominants. So you know that each one contributes 10, 10, 20, 30, 40. So you know that 4 dominant will be contributing 10 centimeters. So you have 40 here. But since you have a base height, because of small a, small a, small b is 10 centimeters, you have to add the 10 centimeter base height. You have 50 centimeters. Okay? Let's go to letter B. Predict the phenotypic ratio of the F1 and the F2 across between small a, small b, and big A, big A, big B, big B. So in this case, you have the parents, homozygote recessive for A and B, homozygote dominant for A and B. So you already know, but just by looking at the F1 would always be a heterozygote. So in this case, what is the F1 generation? All of them will be heterozygote. But what's the phenotype? How many dominant? 1 and 2. And then you know that each dominant produces or adds 10 centimeters. So you have 10 centimeters per A, another 10 centimeters per B, so that's 20 centimeters. But you also know that the base is 10, so you have here 30 centimeters for the F1 generation. For the F2 generation, you will be crossing here two F1s, and the answer that you will get here is what's the F2 generation? Now, to get the F2 generation, just go to the Pascal's triangle. Okay, so how many gene pairs? 1, 2. How many alleles? 1, 2, 3, 4 alleles here. So the equation is A plus B raised to the 4. And you will have a binomial expansion of 1A4, 4, 6, 4, 1. So it's a 1 is to 4 is to 6 is to 4 is to 1 ratio. So that would be the answer to that particular question. Now, if you're going to be asked what would be the genotypes for each of these phenotype, then you just make note of the binomial expansion. So you have A4 here, and A is the dominant. So you have 4 dominants here. 1, 2, 3, 4. And the count is 1. You have here 3 dominants and 1 recessive. So you have 1, 2, 3, 1, 2, and then should be letter B, capital B over here. So 1, 2, 3, and there's two each of these, so you have a count of 4. And then you have here 2 big A, 2 two dominant and 2 recessive. So you have 1, 2, big A and big B as recessive, big A, big A is recessive, big B and big B recessive. So there will be 2 counts per each. 4 of this, 1 of this, and 1 is, so you have a 6 count for this combination of the genotypes. For this one, you have only 1 dominant and 3 recessive. So you have 1 dominant, 3 recessives over here, 1 big B, 3 recessive over here. There's 2 count, there are 2 and 2 there. There are 4 counts here. And finally, all recessive, small a, small a, small b, small b. And that will be how you will be getting the genotypes if it's asked of you. So again, make use of the Pascal triangle, 1, 4, 6, 4, 1. Okay. And list all the genotype that gives rise to plants that are 20 centimeters and 40 centimeters in height. So first of all, how do you get the genotypes for 20 centimeters? You have 1 dominant allele times 10 centimeters, and then the base height is 10, so you have a 20 centimeters. So the 20 centimeters will have 1 dominant allele. For the 40 centimeters, you have a base height of 10, so you have a 30 remaining. So you have 3 dominant alleles multiplied by 10, plus that base, you have a 40 centimeters. So for the 40 centimeters, you have 3 dominant alleles. And that will lead us to this particular genotypes. You have 1 dominant allele, big A here, and all recessive, big B here, and all recessive. Those are the only genotypes for 20 centimeters. The genotypes for 40 centimeters, 3 dominant allele, 1, 2, 3 you have here, and then the other one is 1, 2, 3 dominants. These are the only genotypes for 40 centimeter. Okay, and that ends our discussion for this part. Let's go to the next one.